us once again.
I thank you for having me. It's a pleasure to be here with you virtually. I think this is my first time presenting in Canada virtually. And so I'm being asked to provide an update from a scientific update from the International Celiac Disease Symposium that was held. I have to get to that. That's my last slide. Sorry, I've just stopped screen sharing for a moment.
We're having some internet challenges, so you may have to try again.
Okay. Anyway, I was lucky to have this is probably only my second large meeting that was performed in person. And this was in Sorrento in Italy. See if I can get to.
You're up, you're good.
Good, okay. So the meeting was held in Sorrento. It's the first time that the Celiac Symposium has been held since the beginning of the pandemic. It had been delayed from a previous year. And Sorrento is between Naples and Salerno. It was in person with some parallel sessions, both scientific and clinical. There were minimal to no COVID restrictions that I could perceive. Why in this place? One, it's a beautiful spot if you've never been there, and it's halfway between Naples, where Riccardo Tronconi, one of the organizers, and then Carolina Ciacci from Salerno, which is south of Sorrento. Absolutely stunning place to hold a meeting, and also a center of celiac interest now for more than, I think, 3 generations. The themes that I perceive, yes?
Sorry, we can see your presenter slides. I don't know if you can flip to a mode where you just see the one slide you're on. We see your presenter screen with your next slide coming up as well.
Oh.
Maybe there's display settings.
Is that any better?
No, we're still seeing the same screen. I'm wondering if at the bottom, it's not a giant deal, but just we see 2 screens up, your main screen and the one that's coming up.
Oh. Black. Subtitles. See if I can make a hide presenter view, maybe.
Yeah.
Is that any better?
Just have to give it a second to, there you go. Perfect. You're on your way.
Mm-hmm.
Okay. So the themes that I perceived from the meeting, and some of this is personal, of course, is new discoveries. And we'll talk about the issues of prediction, triggering, and prevention. I'm not going to talk much about diagnosis because we already had an excellent discussion from Ben Lebwall on the issues of diagnosis. And then I'll focus a lot on management. So there was a couple of very exciting sessions on prediction, prevention, and triggering, focusing on genetics and beyond. And we had Iris Junkers from Holland, who really provided us with a 2022 version of the genetics, and it goes beyond genetics to what's called epigenetics. We all have a certain number of genes, probably about between 30 and 40,000 genes. But the subtlety of our genetics is much more determined by epigenetic signals. And some of these are ways that turn on or off gene expression. And those can actually be inherited. You can inherit not only the gene, but some of the epigenetic markers. So things that happen to our grandparents influence health in us. And she was exploring some of these new genes that hadn't really been understood. One thing called LPP she had focused on, showing this is an anti-inflammatory marker within the body, and when it's reduced, we get more inflammation. She also talked about some of these novel enteroids. These are little model intestinal bits, which compose both lining cells and lymphocytes, and that this could be a way to test new treatments in human tissues. And we did an excellent presentation from Calvin Coe from Stanford, who's really been pioneering the uses of these, but they're not the only lab. Daniel Agar, who's a pediatric gastroenterologist from Sweden, gave an excellent talk about this concept of enterovirus infections at around the time of gluten introduction that perhaps this could trigger celiac disease, and potentially this could give us some novel ways to think about prevention of celiac disease in children. I'll show a little bit of that data in a minute. And then Renato Orricchio, a pediatric gastroenterologist from Naples, was talking about biomarker changes in infants who are destined to go on to get celiac disease, but have not yet been even exposed to gluten. And so this is a kind of a concept that you're pushing the idea about what's causing celiac disease to earlier and earlier in development, even things that are happening biologically before the child is first exposed to gluten. Daniel Agar, the picture in the top left from Sweden, and one of the themes of what he talked about was this concept of if the child is getting a lot of gluten and they happen to have certain viral exposures, that seems to greatly magnify their chance of getting celiac disease within the next 10 years, the first 10 years of life. So it's kind of a bad coincidence if you get gluten, high doses of gluten, and you've gotten certain viral infections. And these could be viral infections that are barely noticed in the children. Stefano Guandolini gave an excellent talk about kind of laying out about the issue of diet and celiac disease onset. For example, we just heard an excellent talk about that mentioned fiber as being one of the most favored parts or components of foods for dietitians. Well, a higher maternal fiber intake in pregnancy seems to be associated with a lower risk of celiac disease in the offspring. Avoiding cow's milk baby formulas in early life was not beneficial. However, early introduction of gluten high dose of gluten might be a risk factor, but there is a small so-called EAT study from the US suggesting the contrary. There might be a reduced rate of subsequent celiac disease. So there's mixed data there, and certainly not enough to tell us what to do about gluten in the young life. Introducing gluten in small amounts or delaying its introduction to at or later in the first year of life does not appear to be beneficial in those at high risk of celiac disease. But large amounts of gluten during the first 2 years of life seem to increase the rate of celiac disease. And there was also this perception that a Mediterranean-style diet in the first 2 years of life might be associated with a reduced risk. Lots of caveats, lots of variables here, and so not a lot of certainty, but certainly lots of things to study in the future. There was a very exciting presentation by Francisco Leon, who runs Prevention Bio. That's a company in developmental stage company looking for therapies to prevent diseases or treat diseases, including celiac disease. And he had interesting data on persistence of Coxsackie B infection in patients who later got type 1 diabetes. And as we know, type 1 diabetes and celiac disease are very connected from a genetic risk point of view. And the concept behind this was that perhaps for type 1 diabetes as a model immune disease, that children getting Coxsackie B infection that's persistent, that means they don't clear it, that might be putting them at risk of developing an autoimmune disease. And that company is now developing and even testing a vaccination for Coxsackie B and is undergoing safety trials in humans. And the concept, and this is a concept I would say, is that could vaccination for certain infections prevent later autoimmune diseases? We heard from Ketil Stordal from Norway about that these infections are maybe even competitive, that an infection with one thing might protect you from an infection from something else. And that there's something other infection might be one that triggers disease. So there's a lot of interest about things like season of birth. So for example, children born at a certain time of year might be at increased risk compared to other children, but that's very geographically specific. Luigi Greco, who has spent 30 or 40 years studying gluten as risk, reviewed a lot of what's known about the types of gluten and potentially the risk for later celiac disease. But again, still not a lot of clarity. And Renato Orricchio, who is, I think, a third-generation researcher in celiac, he's a pediatric gastroenterologist in Sweden, in Naples, had presented very interesting data about markers, biomarkers to predict celiac disease before the children ever got gluten. And this is based on children who go on to develop gluten. They've been involved in providing samples early in life. And what we show on this screen is that in black, these are immune markers called cytokines, and they're higher in the children who go on to get celiac disease than those who do not. Now, this is not yet at a stage where you can identify a 4-month-old to say that kid is at risk for celiac disease and this other kid is not. But it sets it up as a potential application of this type of work, that maybe we could start thinking about predicting the risk of diseases. And if we can predict children who are higher risk, maybe then we could come up with strategies to prevent illness in those individuals. I have to come to Canada, and I'm a great fan of Elena Verdu at McMaster, and she and her team had a very big presence at the meeting. She gave an outstanding talk on the role of the microbiota, and a lot of this work was based on comparing healthy controls and celiac disease. She reviewed, a lot of the work that's been done in this field, a lot of which has been done at McMaster, and basically, you're looking at the microbial composition and functions of the bacteria within the intestine. And one of the things that Elena's group have done is they focused on what bacteria are doing, how they interact with the host, that's us, but also how they interact with gluten. And then they've translated a lot of what they see, or what we believe in humans, and translate it to mice and to look to understand more about the interaction. And she reviewed that there's been a lot of work done on typing what are the types of microbes that are present within the intestine of adults and children with celiac disease. And basically this slide illustrates as a huge variety of results. It's very much dependent on what is sampled, but also where the sampling is occurring. Is it occurring in Spain and Italy and Canada and the United States, Mexico, India, etc.? And there are differences that are geographic, and it's trying to sort out which of these are actually associated with disease and which ones are associated with the geography of where somebody is from. But certainly, and for some of these, there were very little changes. For example, here, Canada, US, and Mexico, everything is white. I mean, there wasn't a significant increase or decrease. But in other parts, say Spain and Italy and other places, there were changes seen. So how much of this is real? And it may not be that it's not about what the bacteria are, but perhaps what they do. And that's been a big focus of what Dr. Verdu's lab has done. For example, here is some data suggesting that bacteria interact with the gluten, especially through something called elastase, and that this conditions the gut response to be pro-inflammatory. There may be parts of this interaction where there are bacteria that are good, that tend to maintain homeostasis or regulation, that is not inflammation. And then there is this circumstance of cross-reactivity. Maybe there are things in the bacteria that when we see them, they could potentially be similar to partly digested or altered gluten or gliadin peptides. A tremendous body of work from, and I put it up as my example of outstanding science from the North American continent and from our Canadian colleagues. So she summarized the differences in microbiota depend on the patient, what the study population, the study design and location, and really focusing that we need to be looking at the bacteria in the duodenum. This is where the action of celiac disease happens. And also describing the niche. These are spaces where our bacteria make homes within us, and what they're functional, what are these bacteria doing, and that you can't tell just from looking at stool. And it's this translation, so working between humans, carefully controlled animal models, and then biologic, like cellular testing, that it's across the spectrum. We can understand much more about this choreography of interaction between us, our immune system, our microbes, and what we're eating in terms of driving disease, and encouraging, of course, more collaboration. The next theme is diagnosis, and there was a very interesting roundtable discussion of representatives from around the world, focusing primarily on adult disease. And adults still require biopsies by and large, as we heard from Dr. Lebwohl. Children in Europe and many other places are practicing the biopsy avoidance now in about 50% of children, but they have to meet very specific criteria. However, with COVID, many patients are not getting scoped or even being referred to specialist doctors. Clinical trials are reporting a high proportion of candidates for the trials who do not have properly confirmed celiac disease. And then there was this roundtable discussion about what to do about it in terms of adults. And really one of the concerns they raised was endoscopically, would we be missing things that we'd find endoscopically when we go down to get biopsies? And the data is starting to emerge that really significant things are relatively rare, and that perhaps we could judge who would need to be scoped. And there was concerns raised that maybe patients wouldn't be motivated for a lifelong gluten-free diet without biopsy proof. And also regulations in some countries would require biopsy proof for cost subvention for their gluten-free diet. And they're certainly not relevant in the United States, where there's no cost subvention for patients trying to live on their gluten-free diet. But in general, I think there's it's a slow process of starting to take on board the evolving data about how accurate certain serology can be. And that's we already heard from Ben Lebwall that there's some serology tests that are totally unreliable in terms of diagnosis, and most patients who have positive tests don't have the disease. And I think it's got to be very carefully applied. And the European Pediatric Group, who promulgated this first, made at the very beginning, these children should be referred to a pediatric gastroenterologist to manage the diagnosis. And we've also heard in the question and answer with Dr. Lebwall about what about months of delay that can lead to having a sick person, be it adult or child. And I have to say, as a human, it'd be very hard to keep eating gluten if I'm feeling sick, and this is what I'm told I'm suspected to have. But it does lead to increasing problems with confirming or being able to confirm diagnosis after the fact. The meat of what I want to talk about is management. We'll talk first about diet. There is Nick Trott from Sheffield, who's a real promoter of nutritional intervention in celiac disease, a tremendous speaker, really summarized all of the potential roles that dietitians can have beyond simply monitoring adherence to a gluten-free diet. And we've heard an excellent talk from an expert focusing on the plant-based diet in this context. So there is a whole lot more that our registered dietitians have to offer patients. And we know that management is not just about getting to a gluten-free diet. It's about living well. It's about living healthily. It's about dealing with all of the other issues that nutrition plays. And certainly there was a lot of concern about metabolic syndrome. That's a syndrome of being overweight, excess fat, poor metabolism, hypertension, etc., has been a problem in treated celiac patients, including children. There were a number of presentations about it. The one study I thought was interesting, a flour mixer, how much air does it put into the air? It puts it to about a foot in diameter around the mixer, so around the mixer. So if you're mixing flour in your kitchen, regular flour, well, you have to recognize there's a zone of high concentration that lasts probably half an hour in that. But it's not that the whole house is contaminated. It's a specific area. There was a lot that was done about so-called GIPs, that's gluten intestinal peptides in the urine or stool as a test for gluten, and predicting damage to the follow-up intestinal biopsies. This is especially common in people trying to live with both type 1 diabetes and celiac disease. That it seems that particular population, they're focusing a lot more on their type 1 diabetes as they need to survive, but perhaps ending up with a lot more gluten exposure than would be good for their celiac disease, and pointing out the need for extra support for individuals with that double diagnosis. Also, stool testing is more sensitive than urine testing, and lab testing is probably better than the over-the-counter tests that we already heard is available in the United States. I don't know their Canadian circumstances for that. The gluten-free diet is, at least in Norway, is less nutritious in practice. We've already heard about the importance of fiber, for example, and sometimes the gluten-free diet can be deficient in fiber. Vitamin supplementation or fortification of certain foods that are gluten-free diet may also be an issue. And then several presentations that focused on overweight rates that increase after a gluten-free diet, this one in children from Croatia, but also others. So going on a gluten-free diet is not the end of the nutritional challenges that patients with celiac disease face. What about the satisfaction of people with celiac disease with the gluten-free diet? And this was from Beyond Celiac Disease, based in Philadelphia, and they had surveys over 1,000 people in 2014 and 2022. In that 8-week time period, there was a shift, a downward declining satisfaction with the gluten-free diet as a self-perception of how good is this treatment for me, with a shift to a lot more people were not at all or only slightly satisfied with the gluten-free diet. And of course, that drives the interest in new treatments. And this was a major part of the conference. There were over 24 presentations on novel therapies. 86% of English patients wanted an alternative or adjunctive therapy to a gluten-free diet. There's now 3 big pharma companies and lots of little ones engaged in this field. There were some probiotic studies and probiotics showing degrading of these gluten intestinal peptides, anti-inflammatory bugs, for example. And there were several tolerance trials, which I'll talk about. There's even an egg immunoprotein that's all the way from Alberta, developed by scientists in Alberta. And there's now a phase 2 trial that's ongoing in 2 sites in Canada using an egg-derived immunoprotein. Tight junction regulators, there was some work on a tight junction regulator. Now, larazotide acetate, some of you may be aware of, which is kind of our original tight junction regulator, has just failed its big trial and probably won't be making any more efforts to treat celiac disease. There's work on enzymes that's ongoing, and tissue transglutaminase inhibitors. There are now 3 of them in develop that we'll talk about at the meeting.
Traffic from the blood to the spleen and possibly other lymph nodes.
I'm sorry, what happened here?
And thereby induce tolerance through a variety.
So tolerance methods, there were a couple of different tolerance methods that were discussed at this meeting. One is TAC-101, which is injected. They're little nanoparticles that go to the spleen, and they induce tolerance. And think of it like a turbocharged way of trying to induce like allergy shots almost. You're actually giving little bits of gluten contained within special carriers. And it's the carrier that lets the immune system know that what I'm bringing is a good thing, be tolerant to it. And so it generates tolerance cells shown here in the bottom that then traffic to the intestine. And so there was a suggestion that this might help the intestine. The standard model for a lot of these trials is to take patients doing well on a gluten-free diet, biopsy them, get some baseline bloods, then give them the treatment, and then give them a challenge of gluten. And we'll hear about that with a couple of the other agents. And this is a very standard way. And they want to see, does this treatment in some way blunt, reduce, or stop the reaction to gluten in patients with celiac disease? You know, and after now several decades of preaching about being gluten-free, it doesn't come automatic to be able to tell people to eat gluten as part of a trial, but patients are willing to do this as part of advancing science. And I think it's been quite remarkable about the celiac community is they want to step forward and they participate and partner in moving the field forward. So again, this was a trial, you know, of not a very large number of patients looking at the ability to protect against the effects of gluten. And really what they're looking at in this trial is looking at the ability to block cells that react to gluten. And one can see here in the graphs, in the brown graph is placebo-treated patients. And after a gluten challenge, you can see there's this increase in these cells that are inflammatory. And in blue are the patients treated with the TAC-101, and you can see that that effect is blocked, showing a significant protection from this effect of a gluten challenge. So it seems to be a new approach to inducing tolerance to gluten. It's now starting up, and will soon be starting up into a new trial a much more expanded trial with the sponsorship of Takeda. Before this was a small company, now it's Takeda. And it seems to be, at least theoretically, it seems to have some potential value or promise. But again, this is, I should reiterate, all of these are investigational agents. Nothing is available to prescribe anywhere in the world. And indeed, it's very premature to even think about that as yet. But I think it provides promise in the future of that. The other approach is this. This is a liver targeting therapy. So again, it takes a piece of gluten, connects it to something that brings it to certain cells in the liver. Our liver is our filter for our gut. It's also a tolerogenic organ. And this is another way of inducing tolerance. And there was also a report on this methodology at the meeting. It's a very similar, you give the treatment and then you do gluten challenge. In this case, it's a 3-day gluten challenge, and then look at markers for immune response. And here in the red on the left-hand side, here's the increase in the immune markers that occur with placebo treated. And it seems to be blunted in these other colors, which are treated with the active agent. The next part is this, is the egg. Sorry, I'm having a little technical difficulty here.
A little bit of echoing, yeah. Hopefully it clears up.
Sorry.
Carry immunoglobulins that will bind.
And this is another Canadian study. There was a poster presentation about this. This is, you take chickens, basically immunize them, and their eggs produce antibodies against gliadin. This is then freeze dried and then provided to the individuals to bind gliadin in the gut. And they had shown in an open label study a trend for improvement. And now they're doing a larger study across 2 sites in Canada. So this would be an area we're looking forward to with excitement, a very novel area and certainly scalable. When we think about how many chickens there are, we could generate an awful lot of antibodies. So again, a very interesting and novel approach, but in a very early stage of development. A next approach.
Another approach is the BOT in the New England Journal of Medicine.
The next approach was this treatment of tissue transglutaminase inhibitors. We know that gluten undergoes a reaction that changes glutamine to glutamic acid that makes it very antigenic. And they did this large study, and we heard about this at the meeting around Europe, showing could you give this to prevent the effect of a gluten challenge? And that's exactly what they did.
Sites in 7 countries.
And really it was to try and improve the
measures.
This again is very similar. People put on different doses and then given in this case a 6-week gluten challenge, and then see how patients did with biopsies, etc. afterwards. And these patients
fairly typical
basically showed that there was protection from it. And here, this shows a change in the number of intraepithelial lymphocytes before and after, showing that the patients who were treated with the active drug were protected to some degree from these changes. I wouldn't say that this is a definitive study, but quite a large study. And then this is also going to go into trials, and another large company have indicated their interest in bringing this forward. So it also showed that with the gluten challenge,
you see at the middle time point the increase in symptoms that occur in the placebo treated and those getting the lowest dose of the active drug. And at 2 higher doses, it appears that the symptoms are suppressed.
So moving on, this I apologize. The last one I want to mention is what's called the Celiac Shield study, which was presented, which shows the gluten challenge.
It was an NIH-focused.
And basically,
it sounds like you've got an audio playback or something happening.
I apologize. I'm not sure how I can stop it. basically, this study showed with an enzyme that can break down gluten that you could protect against the damage in the intestine produced by a gluten challenge, in this case, a 6-week gluten challenge. And it also attenuated some of the symptoms that occurred with this gluten challenge. Again, supportive of this as a potential approach. Now, this particular agent has been in clinical trials now for probably 10 years. So I would say it's an effort that's still got some interest, the idea of enzymes to break down gluten. And there was another presentation at the meeting of a bioengineered enzyme called Coumamex that also does something like this, not quite at this stage of development as yet, but this is an area where potentially if someone would take something before eating or being exposed to gluten by accident, that it would protect them from it. But again, all at a fairly early stage of approach. The other thing about this study was the notion that you could test the stool or the urine for these gluten intestinal peptides, and that giving the enzyme to people before the challenge prevented an increase in these urine peptides for gluten, suggesting that it could actually digest a large amount of the gluten that these people were being exposed to. There was very little at the meeting about non-celiac gluten sensitivity compared to previous meetings that had several sessions devoted to it. There's now, I think, an appreciation that most people who think they have non-celiac gluten sensitivity really probably have irritable bowel syndrome with a FODMAP intolerance. Now, that's not all, but it's probably a major part of individuals who feel better when they avoid gluten who don't have celiac disease, at least when it's GI symptoms. There were some work on gluten ataxia, again from the Sheffield group in the UK, talking about tests relating to ataxia, something called tissue transglutaminase 6 antibodies, and potentially a response to a gluten-free diet. There was one study, an Indian study, suggesting that about 12% of people with IBS in India may have non-celiac gluten sensitivity. However, of those, only half of them responded to a gluten-free diet, and only half of those responded with symptoms on re-challenge. They did have shorter villi and microbiota differed in those, so it might not be truly non-celiac gluten sensitivity. There was a New Zealand study suggesting that almost everybody with IBS responded to gluten and dairy avoidance. There was a drug trial of a gluten targeting endopeptidase in non-celiac gluten sensitivity, and it did not prevent symptoms of gluten challenge. So again, suggesting that people who think they have non-celiac gluten sensitivity, probably it's not the gluten. There was another study from Rome suggesting more psychological symptoms in patients with non-celiac gluten sensitivity compared to celiac disease patients or healthy controls. So that finishes my very selective summary of some of the science at the ICDS. The next one is slated to be in Sheffield in the UK in 2024. And we hope there'll be even more advances in celiac science. Thank you for your attention.
All right. Thank you, Dr. Murphy.